and fields, then they created on January the 1st, 2020, a unique institute called INRAE. This INRAE is the French National Institute for Food, Agriculture, and Environment. It is about 10 to 20, I don't remember, 12, I think now we are about 12,000 people, and we have about 300 to 400 researchers all around France. And we have about let's say now 300 locations with about 60 experimental units. And this is about 1 billion euros annual budget, mostly for people remuneration. Okay, we have 18 research centers all around France and 14 scientific divisions, and we work basically on the agroecosystems, animal science, let's say crop and plant science, and we have a number of divisions around ecology, forestry, mathematics, and let's say numerical science and economy. So all fields needed to have a global view on agronomic research in the sense, very large sense. The lab in which I work depends on 3 different divisions that are agroecosystems, transform, and microbiology. So we have basically agronomes, people from biology, and people involved in the transformation of products for food and for environmental engineering. The lab in which I'm working is the LBE, which means Environmental Biotechnology Lab in English. And I lead this European network, Euro-Mediterranean network. The LBE is located in Narbonne, which is a close city, which is a city close to Spain in south of France. On the right, down, you have, on the bottom, you have a view of the lab. It's, let's say, a very nice location. It's very, very pretty, let's say. And the LBE is a world-leading research lab in a biological process that is called the anaerobic digestion process, which is the transformation of organic matter by a microbial ecosystem in the absence of oxygen. And it has the advantage of producing some methane, which is a gas that can be used for producing energy. Okay. This is a view of the lab. Basically, we have 4 research units dedicated to the diversity of, let's say, microbiologists working on microbial ecosystems and what is related to the diversity. We have a team which is called here MaxIP, which is a team working on how putting these ecosystems in work. So let's say this is basically people from chemical engineering. Then I lead a small team on modeling, informatics, system and analysis of models, biological models, which is called SAMI. And we have a team, let's say, dedicated to the how valorizing these research, both to go to the economic actors, so private companies and so on. So valorize the research that we do inside, outside, and then get questions from outside, from private companies, and in order to solve them from, let's say, research point of view. And we have also two other, let's say, locations, both in Narbonne and Montpellier, where we welcome a number of industrial people, so people from economic sphere. In the lab, so we adopt a circular economy point of view, where we try to, let's say, treat the solids, the liquids, and the gas in order to put them again in the cycle of production, transformation, and usages, in order to minimize these, let's say, wastewaters and solid waste, in order for them to adopt these circular economy viewpoint. When we speak about the treatment of waste in general, but of wastewater, because this is much more our history, let's say, we use a number of treatment factories. We process some waste, inputting much energy, very much energy, and then we produce CO2, some sludge. We don't know exactly what to do with that because it contains a number of bad substances, let's say, toxic substances that go back to the environment. And then we have some treated water. From this, let's say, old view of the treatment, we would like to go to the new view of the treatment, which adopt the circular economy viewpoint, where we produce energy from this waste. This waste is no longer treated, but it is processed in a factory that is called biorefinery, where we can minimize the ultimate waste, and at the same time we can produce renewable energy, some treated water that can be used, for example, for reuse, and we produce a large range of added value molecules, both for agriculture, agro-food, and other industries. So we want to go from this view to this one, and then these are the research we are working on. So what's to do with the used water? The idea is to try to reuse it, because this is a very big problem for Mediterranean and Western and Northern Africa. We know that about 60% of water resources are already subject of conflicts in all these zones. And for example, even in Europe, France has refused recently to give some water that is pumped into the Rhone, so close to Marseille. There is the Rhone River, which is a very big river. We pump into it in order to use the water for agriculture, and there is a very big pipe coming until the frontier, but this has been stopped at the frontier, and France refused to give this water to Spain. So even within the European countries, these resources are already subject to conflicts. Okay, this is a very a view that I like, because it shows you the Earth, then the water on Earth. This is this way here, the gutlet. Then you have the fresh water, and then in very small here you have the fraction of this fresh water that human can use. So as you can see, it's quite small with respect to the Earth. So with respect to this equation, there's a very important point, which is that mobilizing new resources is not enough. Of course, we can mobilize new resources as for oil, for example. We find all day, we find new oil on Earth, but for the water, this is the same. We can find new water, but the problem is that its quality is decreasing quite a lot and rapidly, and thus we must be we must take very care of that. And the idea is that we should put our efforts in reusing water, but also in saving some waters. For example, I have data, very new data, very recent data that a professor, a friend of me, gave me. This is Professor Nicolas Roche in Marseille. He explained me the following. Something is said to be sustainable if we can guarantee the functioning over the next 100 years without any additional problems. Today, about 5% only of the water, and it is much less in Europe, it is about 1 or 2%. And it can be very much, for example, in Jordania or in Israel, they use that more than 70%, they reuse more than 70% of their water. But in average in the world, it's not more than 5%. If we can mobilize 40% of wastewaters and reuse these wastewater, this is enormous. Then we report the shortage that we can see of about 40 years only. Now, if we mobilize 40% of wastewater and at the same time we save freshwater withdrawal by 20%, which is not so much, then we report the shortage to 80 years. So it is almost sustainable. So you see that we must reuse, but we must also save water. So what can we do with used water? So we can reuse it, but there are many problems of water quantity and quality. A very recent study of a friend of the lab, a colleague from the lab, show that in France only, about 147 tons of micropollutants, metals, and other toxic organics are rejected into the environment. So we must, of course, reuse water, but be very careful in the way we do it. We must save water and then save our resource and limit the use of these micropollutants in the industry, because the money you earn is before all the money you have not spent. So saving water is also, let's say, should be done in the same way that we can limit the use of these micropollutants. So it means using less substances and less toxic substances in the industry. And then we must, because we must save water also, we can reuse wastewater with appropriate additional treatments to allow a better control of quality, which is rejected finally into the environment, because this is a way of better using the money. So let's go back to the origins of the network. Okay, this is in French, but I can translate these. This is a real change which has been done in INRAE. INRAE is another institute. It's not INRAE. It's the same kind of institute, but related to numerics and the use of numeric science. And they have a very important, let's say, connection historically with Africa. And in the years 2000, a number of people, including Dominique Soto, who was at the head of the connections with Africa, they put the idea that collaboration with Africa must be very developed. And this is for, let's say, the context in which we have created the network. So the first condition for this network to exist is a very, let's say, positive context about the policy of national institute in France in the year 2000. So the protagonists are basically two people which were very involved in Africa in these for many years. Two Fixari who arrived in France in the 90s. He kept a lot of contacts with his university of origin, which was Tlemcen, and they spoke during a number of schools of, let's say, building something positive on this positive context. And then during a school that was held in Tlemcen in 2006, they decided with Brahim Cherki to, let's say, submit a 3+3 project that was a project between 3 countries of Southern Europe and 3 countries of Northern Africa. And they succeeded in that. And then with a number of other people, so a number of people from Tunisia, from Spain, from Italy, and from Algeria, and myself in France. So they decided to create a project then, that was not a network yet, but that was a project around the questions of reusing wastewaters, and then modeling and control basically of processes in order to reuse wastewater. And that was the treasure, treatment and sustainable reuse of effluents in semi-arid climates, that aims at studying innovative integrated solutions for the treatment and the reuse of water. Then, of course, we build on a number of activities. For example, each Sampa school that was organized by one of the partner was the occasion, let's say, for the partners to meet and then to put together let's say the financial support was not so heavy, so was not so important. So we put them in common in order to build that. And then we began, one of the first things that we did is to begin a number of co-advisoring of students on many different research domains, including mathematics, for example, for Miled and Redran, who are Tunisian. Amin Shafi worked on a number of problems, but related to process engineering, and notably the modeling of membrane bioreactors, because membrane bioreactors allows a very advanced treatment, and then there is respect to these pathogens and micropollutants have a very high efficiency. So we worked on these kind of problems. And all other students, such as Walid Bouafs, tried to solve some problems related to optimal control problems. And then in the next years, the objective was to extend the network to other partners. And we took the opportunity, for example, of Mohamed Jawa, who was then in Egypt, working in the French university, in order to include some colleagues of him. Then we extended the network into Morocco, into Greece, and then worked all together with bilateral projects or European projects. It should be said here that the network at that time, it moved because the initial projects ended. So at that time, in about 2010, the project's treasure then moved into a network, which is much more, let's say, moving over the time, because some partners come and some partners go out, but it's much more moving always. And then we had the opportunity with a great people who is Bernard Philippe. He had a very interesting idea, which was to put all together all the partners involved in the 3+3 INRAE project. He decided to put them all together with their partners, of course, of the North and of the South. And he submitted a European project, which is specifically dedicated to the exchange of students. This, if I remember well, this instrument of Europe does not exist anymore, but there are new instruments that exist today. But at that time, that was very interesting because it helped us to co-advise a number of PhD students within our treasure network. And then we also took the opportunity of bilateral projects where, for example, Brahim Cherki got... Yes, is there any question, or maybe someone has in microphone open? Okay, I continue. Okay. And then we began to apply for some bilateral projects, such as this one, which was dedicated to the treatment of wastewater with membrane bioreactors. It was with France, Algeria, and Senegal. And it was quite important because we got 55,000 euros for a number of experimental device missions, and of course personnel on the modeling, the control, some experimental work, and some mathematical ecology. So as you see, it's just examples of this kind of project, but of course we applied for others and got others, and so on. So what do we do? I will show a number of examples, but before I will show you, let's say the idea in order to make these circular economy viewpoint a reality. So we begin by what the available crop plant models tell us about the needs of plants. What they tell us is that if you look over the time at the needs of the plants, their needs in terms of their basic resources that are nitrogen, water, and phosphorus, these needs over time are very varying, and they are, let's say, decoupled one from each others. The idea, the actual idea of reusing water is that you treat water first. So you remove everything which is inside the water, then you transport this water and you store it, and then you use it in the field. But the problem of that is that you eliminate a number of nutrients that are present in wastewater, but they are beneficial to plants and crops. And then this question, why eliminating nutrients from wastewater while they are beneficial for plants and for crops and for agriculture in general? So the idea is to implement a number of sensors directly related to some usages. You can refill reservoir, you can, for example, use a number of sensors that inform you about the needs, the online needs of plants and soils, then get this state and the needs of the plants to the treatment plant, which is now a biorefinery, if I can remember you. Then you have, in the inputs, you have a number of, let's say, characteristics of the wastewaters in terms of flux and in terms of qualities. And you have a number of disturbances regarding the weather conditions. And if you look this scheme, this is a very classical control scheme in control theory. So the idea is, can we develop researches around the use of modeling and control in order to specifically adapt the needs of the plants and on the usage in general, using the available knowledge, and then uh, feedback these informations with respect to a number of inputs, some of them being known, some of them being unknown, and under a partial and a disturbance, let's say, environment. So this is classically a control problem. Right now, regarding the, let's say, regulations in many countries, it is not allowed to keep these nutrients. But it's not, let's say, a big problem in terms of ID, because it means that you remove these nutrients and then put them back afterwards, after you have transported these water, and you put it afterwards in adapting their quantity to the needs. But the main idea remains, let's say, good and true, which is adapting what the plants and the usages needs in order to optimize the loops. So whatever the objectives, whatever, let's say, the way you do it, the objective remains the same: adapt the quality and the quantity of water to the needs. Okay, in order to go a little bit more in details, may I ask everybody to close the microphone? Okay, thank you. Okay, this is an example of some work that I've been doing, and that will give you, let's say, a little idea about the kind of mathematics we are doing in the network. This is a work related to the use of anaerobic digestion process. So let's say, for example, this system here may be an aerobic system or an anaerobic system. In any case, the idea is to treat the water, and then okay, and then to change the paradigm and then use this biorefinery with a specific kind of, let's say, substrate resource, which is microalgae. Imagine you can use microalgae to treat the wastewater, then you produce a biomass, which is a solid biomass, which is specifically microalgae. And then you have captured some CO2, you have changed, let's say, the kind of, you have put the pollutants into a solid form. They have incorporated, let's say, everything which is toxic in the water. And then you can use these microalgae as a resource in the anaerobic digestion process. So when I say that this system here is, in fact, the coupling of two systems: a treatment plant using microalgae, where you use the pollutants in order to make the microalgae growing, and then you use the output of this system, which is basically some solids, specifically the microalgae, and you digest them in an anaerobic digestion process in order to produce some biogas. Okay, so this system can be, let's say, formalized with a two-step system, where a first microbial ecosystem transformed these microalgae into an intermediate, which is called here VFA, which is volatile fatty acid, but it's not important. We have, let's say, an intermediate substrate resource, and then this resource is transformed by another microbial ecosystem into biogas. So we have two steps, okay, in this system. Using, let's say, classical models, I will not go into the details, but using classical models, we can model this biological system, and our objective is to better know this system. So in particular, we characterize their steady state and the nature of these steady states in terms of stability, notably. So for example, you have a first table here, which just as a function of the functioning parameters, which are the inputs. So for example, the volume of the system, the fluxes going into the system, the characteristics of the inputs, and so on. So we can characterize the steady state of the system. And the other table is about the nature of these steady states in terms of stability. And using this information as a function of the inputs, we can, for example, plot what we call a, an operating diagram. I explain to you what it is. You see that here, depending on the nature, on the stability nature of the steady states, we can define a number of areas which are defined as a function of the inputs. So let's say as a function of the inputs, there exists, these defines a number of areas in the plan of the two controls. So the two controls, excuse me, are the inputs of the system, which this is the concentration in the input, so in the microalgae, and this is the flux at which these inputs is going to the system. So we have two inputs. As a function of these inputs that are called S in and D here, the different areas define the kind of the number of steady states and their stability in this scheme. So it means, for example, that if you are in this region here, which is called J4, then if I go here, I know exactly who is existing in terms of steady states, and I exactly know, let's say, the nature of these steady states. So for example, in J4, if you look at what is written here, I see that there is only one S, which means that there's only one equilibrium which is stable, and all the others either do not exist or are unstable. So it means that if I use any inputs corresponding to this first input here and this second input here, so a set of inputs in this area, I know that there is only one stable point, and I am able to characterize these. So it helps, let's say, the user, the experimenter, to know exactly what are the steady states that exist as a function of inputs. And for example, this highlights a very interesting, let's say, characteristics of this system. For example, here, I fix the input here at about, let's say, 17, and I increase the here the flux. So this is the flow rate. You see that from this point here, I will go into J6, where J6 here is an area, a region of B stability. You see that there are two stable equilibriums. One of each is not expected, but you see that if I increase a little bit more the inputs, I go back in the blue region. So it means that in increasing the flow rate, I can stabilize the system, because in the blue, I recall you that asymptotically there exists only one equilibrium. So this was the first time these characteristics were highlighted. So it gives you an idea of the kind of knowledge we produce in the network and how this network can be used by experimenters. Okay, so now I will give you, before finishing, I will give you a number of open problems we work on. Then it will give you a, let's say, a view of these problems we are working on. So One of the problems is to model and to develop sensor for plants. How can we, I told you we must know the state of the plants and of the soils. The problem is that today there are very few sensors able to tell you if a plant needs some water or some nitrogen. So this is the first work we have to do, linking, develop new models first, and then linking all what we can get in terms of sensors in order to get the state of the plant. We must, of course, uh, install, let's say, validate these sensors and validate models and develop these sensors in order to get an idea of the water available for the plant in the soil. And we must also think of the representativity of these sensors in a large scale, because when you want to control, of course, you will not put one sensor for one plant. So you must understand how the field is it can be viewed in terms of water and available nitrogen in all these in let's say at a spatial to to be representative, let's say, of a spatial heterogeneity. So these are the kind of work we are doing. In terms of modeling and engineering, so this is in French, but I can very easily explain to you. It is related to the additional treatment we must do in order to avoid pathogens and micropollutants in the water. Um, One of the idea is to provide, let's say, to keep the good nutrients in the water while eliminating these toxic substances like pathogens or micropollutants. And one way to do that is to use membrane bioreactors, because the physical membrane allows us to separate this toxic matter from the water with its nutrients. So we must work on the optimization of this MBR, because the presence of the membrane is very costly in terms of the economy of the process. It is very costly to maintain an important pressure on the two sides of the membrane in order to separate the solids from the liquid. So this is a big problem. Uh, this is also in French, but it simply says, let's say, it's a list of the kind of problems we have to deal with. Because we work with bioprocesses, we basically work on the modeling. So we must analyze, simulate, make the links with chemical engineering. We must make the links with microbiology, and we must make the links with control engineering and applied mathematics and optimize the whole system. So this is about what we call the flexibility of treatment systems for the reuse. So we have a number of PhD theses now working on these different problems. So I will not go into these ones because this is just this is related to the way we can use the transport pipes between the biorefinery and the plants. We must use pipes in order to transport the water. And one idea is to consider this pipe as part of the biorefinery. So it means that the biofilms, which are microorganisms, which develops along these pipes, could be used as, let's say, additional treatment to remove either some pathogens or some micropollutants from wastewaters. Because when you go from the biorefinery to the crops, it takes sometimes hundreds and hundreds of meters. So this is an additional, let's say, bioprocess that we must understand. And the spatial heterogeneity of this system is fundamental. So we have now to couple these transport, let's say, phenomena with the biological phenomena and model them and use them in control system, control loops. And finally, we have problems, of course, of mixing different qualities of water in order to them to be coupled with model of plants and then develop optimal control in order to minimize the quantity of let's say nutrients that will finally will go in the soil without being used by the plants, and these must be minimized. So let's go to the conclusion. So we are working on the bioprocesses, and we try to optimize them in terms of uh, using modeling, let's say, and control tools. We we showed that theory can help experimenters. I showed you an example to better understand the process. We must optimize what is, let's say, what we can model, so microbiology, and all the results are usually theoretical, but we work with people from uh, let's say, microbial engineering and process engineering in order to validate them. I would like to add that it is not hazard if the LBE is, let's say, leading this network. This is because the pluridisciplinarity is really at the heart of this Euro-Mediterranean network. And the idea is that people from a given field begins to think as if it was specialist in another field. So, for example, the people who are mathematicians and who work on, let's say, microbial ecology problems, then they begin over the time to think as if they were biologists, and then they can speak with biologists and then understand, they can understand themselves. So these are new questions we will go towards in the next, in a number of actual PhD and in the next PhDs. We go towards the use of reduction techniques, model reduction techniques, in order to try to make the links between very complicated models that exist usually in different fields like chemical engineering, but these models are not useful for optimization. So if we can reduce them, then we can apply a number of optimization techniques. We go towards a number of optimal control design. With respect to these simple models we try to develop. And we work on the optimization of the MBR systems because they are very good candidates for advanced treatments of these bioprocesses for reuse that we plan to do. This is a very rapid historical perspective for Treasure. It gives you, let's say, the list of the production that at a very, let's say, global scale. This will be the 50th anniversary of the network next year. It's about 15 labs that are actually interacting together. Of course, it depends on ongoing projects and affinities. We had 8 bilateral projects, 1 European project, 25 PhDs, about 50 international papers in journal papers. There was a UNESCO chair working in ENIT in Tunis until 2012, if I remember well. we obtained a new project in order to, let's say, make it coming back next year. So there should be a new chair there. And we have organized a number of international schools, notably with Sympa. It's more than 2 actually, but I don't remember exactly the data, but Nabil should have it. And I would like to insist on the fact that most important than all, a very important lever is to make the network living. So it means that we must take a number of actions and animation all let's say all years. We must work on that, and each year there is a 3 full day annual meeting where all people can meet all together. And finally, of course, you need a nice bunch, let's say, of friends. Without nothing would have been possible. Okay, you can get more information on the web, and I remain available for any information and question. Thank you for your attention. I hope I have not been too long, but I have tried to give you an idea of the activities of this network. Thank you very much, Nabil, in any case.
Thank you, Jerome. If there is some questions?
If there are questions afterwards, no problem, you can take contact with me.
Okay.
Fathi? Yes, salam alaikum. How are you? Thank you for a nice lecture. Can I ask the name of the diagram that you had before?
What is the diagram?
The name, yeah. What did you call that diagram? The one that has the colors, the different J's, etc. Le nom de la figure. C'est quoi les diagrammes?
Mais laquelle figure? C'est ça que je ne comprends pas.
La figure avec les diagrammes avec le G4. J4. J4.
Le diagramme opératoire, Jérôme.
Ah, pardon. Ok, excuse-moi. Oui, this is what we call the operating diagram. There has been a paper published in the 17, I think, and it gives. Okay, yes, this is this one. Okay,
this is it. Yes, so it looks like a bifurcation. It's almost like a bifurcation type of diagram. You know,
yes, if you go towards, let's say, these red line, for example, yes, you follow exactly a bifurcation diagram. Here, you have the total view with all these the inputs. That's the difference. Yes, you're right. The link with the bifurcation diagram is that you go here on one line on this side or on the other. Yes, as a given parameter of the system.
That's very nice. I really appreciate it, and thank you for a very nice lecture. I would like a copy, if possible. Professor Nabil, maybe he could.
Yes,
send us a copy or something.
I can share this version. Fethi, I will add a number of